Hello everyone. I welcome you all to Biology Lessons with Kalyani. In today's class, we will be discussing about respiration in plants. The different topics which we will be covering today are what is respiration, glycolysis, Krebs cycle, electron transport system, and finally we will be talking about respiratory quotient. Now, we all know that living organisms, be it plants, animals, or fungi, they need food for their survival. Now, even if we see plants which can make their own food, they have non-green parts to which food must be translocated. In animals, we have herbivore, which feed on plants, carnivore, which feed on the animals, and we have fungi which feeds on dead and decaying matter. So indirectly, if we see that whatever food the living organisms are uptaking, they are directly or indirectly produced by photosynthesis. And this food, it is broken down and energy is released for which is utilized for different life processes. Now the process in which the food material is broken down and energy is released is called as cellular respiration. Is that clear? Now this energy which is released in respiration is not just released as heat, but it is trapped in the form of high energy molecules or ATP. So when I say in respiration, the food is broken down, it means essentially that the complex compound or the carbon-carbon bonds of the complex compounds are oxidized in the process of respiration. So whatever organic compounds undergo this oxidation, they are called as the respiratory substrates. The common respiratory substrate for respiration is carbohydrate, which is used most commonly, but proteins, fats, and organic acids can also be used under certain conditions, right? Now, coming to prokaryotes and eukaryotes, what is the difference uh, with respect to the site? Now, prokaryotes, they do not have mitochondria. So, essentially, the respiration, it takes place in cytoplasm. And if you see the type, it does not need oxygen or it is anaerobic in nature. Now, if you consider the eukaryotes or the higher organisms, respiration takes place in cytoplasm as well as the mitochondria, and it is aerobic in nature. Coming to the energy release. So this energy is which is produced in the process of respiration. It is not released in a single step. But how is it released? It is released in slow and stepwise reaction. And how is the energy trapped? It is trapped in the form of high energy molecule, which is ATP. That's why it is also called as the energy currency of cell. Now, the energy released in oxidation is not used by the cell as in how wherever the energy is released, but it is stored in ATP in the form of ATP, and whenever the cell needs that energy, it will convert the ATP to ADP, and the energy can be used to drive the cellular functions. Apart from the catabolic aspect of respiration, there is also an anabolic aspect, that is, in the process of respiration, different carbon skeletons are produced. These can be used to biosynthesize other molecules. That's why respiration can be considered as both a catabolic pathway as well as an anabolic pathway. So it is often referred as an amphibolic pathway. Is that clear? So these are the different important points which you need to remember about respiration. So if we consider the plants, respiration takes place in them, but unlike animals which have an elaborate respiratory system, each plant part can take care of its own respiratory needs. How? If we see the plants, the leaf, okay, they have minute pores which are called as stomata. These minute pores can open up and close and can help in gaseous exchange. If we see the young stems, they have also the stomatal openings. Now, in case of the woody stem, they have a layer of bark underneath which they have the primary tissue, and then the secondary tissue inside consists of the pith, which is dead in nature and is helpful for the mechanical support. So these barks, they have minute openings in them. These minute openings are called as lenticels, right? Now, so that is how the stems, the woody stems can take care of the gaseous exchange. Clear? Now, if we see the roots, they have root hair. These root hairs, they are in contact with the soil which have air space in them, and the root hairs can take up the air from these air spaces in the soil. So you see, each plant part is able to take care of its respiratory needs. So the gases exchange can easily take place with the help of diffusion, because the gas or the oxygen, it does not need to travel a lot of distance in case of the plants. That is why the respiration rate in case of plants is slow. Okay, and plants do not show greater demands for gaseous exchange. Also, if we see the plants, they in the daytime, they release oxygen in the process of photosynthesis. So that is how they can meet easily the oxygen demand required for their respiration process. Now, if you see in the night time, plants need to take up the oxygen atmospheric from the atmosphere. That is why we are generally not advised to sit under a tree in the night time, right guys? And another important feature seen in plants is that plants also have a loose packing of the parenchyma cells present in them, which has a lot of air spaces. So you see, each plant part is directly or indirectly in contact with the air surface. So the respiration or the uptake of gases can take place easily, and they do not need any specialized respiratory systems in them. I hope that point is clear now. If you see aerobic respiration, in case of that, complete combustion of glucose takes place. So it is given by the equation C6H12O6 plus 6O2 plus gives rise to 6 molecules of carbon dioxide, 6 molecules of water, and energy which is stored as ATP. Now, the respiration, it always does not take place via the aerobic route. If you see the anaerobic respiration, partial oxidation of glucose take place. This process in which partial oxidation of glucose takes place is called as glycolysis, in which glucose is converted into pyruvic acid. Now let's see the process of glycolysis in detail. Glycolysis is derived from the Greek words glykos, which means sugar, and lysis, which means splitting. Now, this scheme was given by Emden, Meyerhoff, and Parnas. That's why it is also called as EMP pathway. Now, if you consider all the living organisms, glycolysis is the common pathway in them, and prokaryotes or the anaerobic organisms, the only process of respiration found in them is glycolysis. Clear? Now, where does this take place? If you see the prokaryotes, they lack mitochondria. So essentially, the glycolysis, it takes place in the cytoplasm of the cells. Okay. Now, the glucose, it undergoes the partial oxidation and results in the formation of two molecules of pyruvic acid. If you consider the plants, they have the glucose which is stored in the form of sucrose and storage carbohydrates in them. So how do they undergo glycolysis? So here what happens, they are converted, sucrose and the storage polymers, they are converted to glucose and they can enter the glycolytic pathway. Okay. Now fructose, if you see, fructose is also found in plants. So what happens to it? Fructose also undergoes glycolysis, but there will be 9 steps in the glycolysis of fructose instead of instead of 10 which is found in glucose. We will be looking into it more in detail. Now let's see the steps of glycolysis in the next slide. This is the schematic representation for glycolysis, and as you can see, we are having 10 steps here. So glycolysis is a 10-step reaction, and in this, one molecule of glucose is being converted into two molecules of pyruvic acid or pyruvate, right? Now we have some steps in glycolysis in which ATP is being utilized, that is, energy is utilized. So it is step number 1 and step number 3 in which ATP is utilized. And there are some steps in glycolysis in which energy is released in the form of ATP. That is step number 7 and step number 10. We have energy which is released as ATP. So this release of energy as ATP is called as substrate level phosphorylation. Okay, and in step number 6 we have NAD plus, which is getting converted into NADH and H plus. So let us see glycolysis in more detail along with the enzymes. Let's go to the next slide. We saw that in glycolysis, in some steps, energy was consumed in the form of ATP, and in some, energy was released in the form of ATP. So we will divide the glycolytic pathway into energy consuming stage, in which we will have the step 1, 2, 3, and the steps 4 through 10, they will come under energy releasing stage. Okay. So the one molecule of glucose, it utilizes a molecule of ATP and gets phosphorylated to glucose 6-phosphate. The enzyme hexokinase catalyzes this reaction, and this step is called as glucose activation step. Okay, now this makes sure that glucose enters the glycolytic pathway. So this is the first irreversible step of glycolysis. So I have marked it in green. Okay, now in the second step, glucose 6-phosphate is converted to its isomer fructose 6-phosphate, and the enzyme responsible for it, phosphoglucoisomerase. Again, in the third step, which is also an irreversible step, one molecule of ATP is utilized and fructose 6-phosphate is phosphorylated to fructose 1,6-bisphosphate. Okay, now this step is very important and is called as the pacemaker step of glycolysis, and the enzyme responsible for it is phosphofructokinase, okay, or PFK. So step number 1 and step number 3 are very important steps, and they are the irreversible steps of glycolysis. Next, we have the second part in which energy or ATP is released. Okay, so one molecule of fructose 1,6 bisphosphate, which is a 6 carbon molecule, it is converted into two 3 carbon molecules, that is glyceraldehyde 3 phosphate or PGAL, and dihap or dihydroxyacetone phosphate. Okay, so these 3C compounds, they are interconvertible, and the enzyme responsible in this is isomerase, triose isomerase enzyme. Okay, now by itself, dihap cannot undergo the subsequent stages of glycolysis. It needs to be processed or converted into glyceraldehyde 3-phosphate in order to undergo the subsequent stages. Okay, now one molecule of glyceraldehyde 3-phosphate or PGAL, it is in the sixth step converted into 1,3-bisphosphoglyceric acid. This step is very important. Just pay attention here. See what is happening. So first glyceraldehyde is getting oxidized into glyceric acid. Okay, and what is helping in the oxidation process? NAD plus is getting reduced to NADH and H plus, and this helps in the oxidation of the glyceraldehyde to glyceraldehyde 3-phosphate to phosphoglyceric acid. Another thing which is happening is addition of a phosphate group, but we don't see any ATP which is added here. So the phosphate group is coming from the inorganic phosphate, okay, Pi, or the H3PO4 in the solution. Okay, so essentially in step number 6, with glyceraldehyde 3-phosphate, there is oxidation taking place in the first stage, then addition of the phosphate group is taking place. Okay, then in the seventh step, BPAG or 1,3-bisphosphoglyceric acid is converted into 3-phosphoglyceric acid. So what is happening? One phosphate group is lost. And who takes up that phosphate group? ADP is taking up that phosphate group and ATP is formed here. Okay. So this is the first reaction in which substrate level phosphorylation is taking place. Now, you should note here that from one molecule of PGAL, one molecule of ATP is getting formed. But we have how many? Two molecules of PGAL. Why? Because this dihap also gets converted into PGAL. So essentially in this step, there are two molecules of ATP which are generated, or these two molecules of ATP are produced by substrate level phosphorylation. Okay. In the eighth step, the 3-phosphoglyceric acid is, there is a change in the position of the position of the phosphate group. So it is converted into 2-phosphoglycerate or 2-phosphoglyceric acid, and the enzyme responsible for it is phosphoglycerate mutase. Okay. Now, this 2-phosphoglycerate, it is converted into phosphoenol pyruvate, and this step, the water is released. Okay. Now, the enzyme enolase is responsible for this reaction. Okay. Now, 10th step is also the another important step in glycolysis. It is also irreversible in nature, and here again a molecule of ATP is formed. So when phosphoenol pyruvate is converted into pyruvate, one molecule of ATP is formed. But we have two molecules of uh, glyceraldehyde phosphate which are undergoing the reaction. So we will be having two molecules of phosphoenol pyruvate which will be making two molecules of pyruvate. So we will get two molecules of ATP here. Again, we will have two ATPs which is coming from substrate level phosphorylation. The enzyme catalyzing this reaction is pyruvate kinase. Okay, so look here, in the energy consuming stage, we are utilizing two molecules of ATP, and in the step number 7 and step number 10, we are having two molecules of ATP produced. So 2 plus 2 makes it 4. So total net gain of ATP comes to 4 minus 2 which are consumed. So that gives 2 net ATPs which are produced by substrate level phosphorylation in glycolysis. Is that clear? So see here, step number 1, 3 and 10 are the important irreversible steps of glycolysis. Okay. So that's all about the steps of the glycolysis and the various enzymes in it. We will just cover a little more extra about step number 6. So let's look at it now. Let us now see how many redox equivalents are formed in glycolysis. Okay, for that you need to see step number 6 of glycolysis, where glyceraldehyde 3-phosphate is getting converted into 1,3-bis-phosphoglycerate. So essentially what is happening here? In the first step, glyceraldehyde 3-phosphate is oxidized by the enzyme and it is forming the glycerate or the acid from the aldehyde. Okay. Next, it is transferring the hydride ion to NAD plus. Okay. Now look, this is the this is the enzyme glyceraldehyde phosphate dehydrogenase, and this is its active site which is having cysteine and histidine residue, and this is the site for the cofactor where NAD plus is binding. So as the substrate glyceraldehyde 3-phosphate binds to the active site, it is getting oxidized and subsequently it is transferring a hydride group to NAD plus, which accepts the H minus ion and is neutralized to NADH. Okay, meanwhile one H plus is also released. So what is a hydride ion? Let us see. Hydride ion means when a proton takes up two electrons, it acquires an extra negative charge and becomes H minus. So here the proton and the two H plus and the two electrons or two E minus can be referred to be as redox equivalents. In other way, the NAD plus, which is uptaking this hydride ion, it becomes NADH, and also one extra H plus is released. So these NADH and H plus can also be considered as the redox equivalents formed in glycolysis. Okay, so how is this H plus formed? When glyceraldehyde 3-phosphate transfers the H minus ion, so it is essentially transferring one proton and two electrons. So here the charge is becoming negative and is unstable. So the solution, it transfers one H plus ion so that it becomes neutral. That is why when NADH is formed, an additional H plus ion is also found released along with it. So as we have one molecule of glyceraldehyde 3-phosphate which is forming one molecule of NADH and H plus or two redox equivalents. So when we have two molecules of glyceraldehyde 3-phosphate in glycolysis, we will have a total of 2 cross 2 or 4 redox equivalents formed in glycolysis. I hope this point is clear now. Along with that, you should also know that after the oxidation of glyceraldehyde 3-phosphate, there is also an addition of the phosphate group, right? Where does this phosphate group come from? It comes from the inorganic phosphate or the H3PO4, and ultimately 1,3-bisphosphoglycerate is formed. Is that clear? This is a very important concept and I hope you are clear with it. In the last slide, we saw all the steps about the glycolytic pathway, and we saw that the end product of glycolysis is the pyruvic acid. Next, what happens to the pyruvic acid in respiration? Is it the final product or it is processed further? So we have two situations here. If we take the aerobic organisms, the pyruvic acid is completely oxidized and undergoes a Krebs cycle. And if we have the prokaryotes or some unicellular eukaryotes, what happens in them is we will have the anaerobic respiration or fermentation pathway, in which pyruvic acid gets converted into either lactic acid or an alcohol. Now these fermentation pathways, they are the process of incomplete oxidation and is generally seen in the prokaryotes and some unicellular eukaryotes. In case of fermentation pathway, after a common glycolytic pathway, the pyruvic acid, which is the end product of glycolysis, can either lead to the formation of lactic acid, which is seen in some lactic acid bacteria, as well as the muscle cells during exercise conditions, where the pyruvate, the 3-carbon compound, is reduced to lactate, which is again a 3-carbon compound, utilizing a molecule of NADH, which is oxidized to NAD+. So essentially, reduction of pyruvate to lactate takes place in lactic acid fermentation. And the net release of ATP is 2 ATP, which was generated in the process of glycolysis. Alternatively, the pyruvic acid can undergo alcohol fermentation, which is seen in the case of yeast. And here, the pyruvic acid, which is a 3-carbon compound, is first decarboxylated, and we get the formation of acetaldehyde, and the final product, ethanol, is formed by the reduction of acetaldehyde utilizing a molecule of NADH, which is oxidized or converted to NAD+. So in alcohol fermentation, we'll have a decarboxylation step catalyzed by enzyme pyruvate decarboxylase, and a reduction step which is catalyzed by alcohol dehydrogenase. So main difference between lactic acid fermentation and alcohol fermentation is the carbon number does not change in lactic acid fermentation. No carbon dioxide is formed here, whereas in alcohol fermentation the carbon number is reduced or a 2-carbon compound is formed, and decarboxylation or production of carbon dioxide, release of carbon dioxide, is seen in alcohol fermentation, which is not found in the lactic acid fermentation. So both these fermentation pathways are the partial oxidation of glucose, and they take place in the cytoplasm and do not need the oxygen for the purpose. Is that clear? Now next, let us look at some important points about fermentation. So fermentation, where does it take place? It takes place in prokaryotes and some unicellular eukaryotes such as yeast. Now we already saw that alcoholic fermentation, it takes place in yeast, and lactic acid fermentation in lactic acid bacteria as well as the muscle cells. Now the essential steps of alcoholic fermentation are glycolysis, followed by decarboxylation and reduction. Whereas in lactic acid fermentation, we will have glycolysis and reduction process. Okay. In both the fermentation pathway, the reducing agent which is used is NADH plus H plus. Okay, and 2 NADH are reduced to 2 NAD plus in both alcoholic and lactic acid fermentation per glucose molecule. Why? Because one glucose molecule produces 2 pyruvate. So from one pyruvate we were reducing 1 NADH. So for a molecule of glucose, 2 NADH will be reduced in both type of fermentation reactions. Also, per molecule of glucose, how many ethanols will be released? So we will just multiply it by 2. So for one pyruvate, we were getting 1 ethanol and 1 carbon dioxide. So for a glucose, we will be getting 2 ethanol and 2 carbon dioxide for alcoholic fermentation. Similarly, in lactic acid fermentation, for a molecule of pyruvate, we were getting 1 molecule of lactic acid. So for a molecule of glucose, we will have 2 pyruvates, and so 2 molecules of lactic acid will be formed. So I already told lactic acid fermentation does not involve the decarboxylation process. So no carbon dioxide is formed in the lactic acid fermentation. In both the types of fermentation reaction, the common reaction pathway is glycolytic pathway. Okay, if we have to talk about the net gain of ATP in fermentation, it is 2 for the both the types of fermentation pathways, and the 2 ATPs come from the glycolytic pathway. Okay, now in both lactic acid and alcohol fermentation, the energy which is released is very less. It is less than the 7% of energy in glucose, and less than 7% of energy is trapped into the ATP. Okay, both the processes are hazardous in nature and take place in cytoplasm and anaerobic condition. Now, it has been seen that as the process is hazardous, yeast poison themselves to death when the concentration reaches about 13%. So, maximum alcohol concentration which can be obtained naturally is 13%. So, higher alcohol concentrations, if we want, we can obtain it by distillation reaction. So, that's all about the fermentation pathway. I hope you are clear about the concepts in this. So until now we covered the glycolytic pathway as well as the fermentation pathway, which were the incomplete oxidation of glucose. Let us see the complete combustion or oxidation of the glucose, which is the aerobic respiration pathway. Okay. Aerobic respiration pathway leads to a complete oxidation of organic substances in the presence of oxygen and releases carbon dioxide, water, and a large amount of energy. Now, where is this aerobic respiration pathway seen? It is seen in the higher organisms. Essentially, aerobic respiration has 4 steps in it. The first step is glycolysis, which is a common pathway. Second, we have something called as the link reaction. Third step is Krebs cycle. Fourth is the electron transport chain, or the ATP synthesis step. Now, where do these particular reactions take place? Now, aerobic respiration can take place in the aerobic bacteria as well as the higher eukaryotes. Now, in case of aerobic bacteria, glycolysis, link reaction, and the Krebs cycle, it takes place in the cytoplasm, whereas the electron transport chain, it takes place in the plasma membrane. Now, if you consider the higher eukaryotes, the glycolysis and the glycolysis takes place in the cytoplasm. Both the link reaction and the Krebs cycle, it takes place in the matrix of mitochondria, and the electron transport chain, it takes place in the inner membrane of mitochondria. Okay, so here the complete combustion of the pyruvate in aerobic respiration leads to the formation of, for a single molecule of pyruvate, we will have 3 molecules of carbon dioxide. So glucose leads to 2 pyruvate. So 6 carbon dioxide are produced because of the complete oxidation of glucose. Now, what happens, the electrons which is removed as a part of the hydrogen atoms are after the pyruvate is formed in the glycolysis cycle, it undergoes a step before entering the Krebs cycle. That step acts as a link between the glycolysis and the Krebs cycle, and it is called as the link reaction. This takes place in the mitochondrial matrix, and typically here pyruvic acid, which is a 3-carbon compound, undergoes a undergoes an oxidation step in which the H group is released and reduces the NAD plus to NADH and H plus. Subsequently, the carbon dioxide group is released. So, a decarboxylation step takes place, converting the pyruvic acid, which is a 3-carbon compound, into acetyl coenzyme A, which is a 2-carbon compound. Essentially, the acetyl group, which is obtained after the release of the carbon dioxide, it is attached to the coenzyme A, and this particular 2-carbon compound, acetyl coenzyme A, it enters the Krebs cycle. So essentially in the link reaction, a step of a step of oxidation and decarboxylation takes place. So link reaction has oxidative decarboxylation that takes place, and this is accomplished by the enzyme pyruvate dehydrogenase, which has both carboxylase activity as well as the reduction activity. Now it needs magnesium 2 plus ions for its enzymatic activity. So that's all about the link reaction. Okay, now let's move on to the Krebs cycle. Let us now see the step 3 of respiratory cycle, or the Krebs cycle, or TCA cycle, okay, otherwise known as the citric acid cycle. So this particular cycle was called citric acid because it was the first product formed in this cycle, and it is also referred as Krebs cycle because the scientist who first elucidated it was Hans Krebs in the flight muscle cells of the pigeon. Okay, so what essentially happens in Krebs cycle? Now, the acetyl coenzyme A, the 2-carbon compound, which was formed in the link reaction in the mitochondrial matrix, it undergoes a condensation reaction with 4-carbon compound, that is oxaloacetic acid. Because the condensation reaction takes place, the carbon compound formed is a 6C compound, that is 4C plus 2C gives a 6C compound in a condensation reaction. Now the enzyme catalyzing this is citrate synthase, and the citric acid hence formed undergoes 2 steps of decarboxylation or removal of carbon dioxide, and there are 3 steps of NAD plus reduction being converted into NADH and H plus, and 1 step of FAD plus reduction converting to FADH2, as well as there is a substrate level phosphorylation leading to the formation of GTP from GDP. So these important steps take place: two decarboxylation step, three steps of reduction of NAD plus to NADH, one step of reduction of FAD plus to FADH2, as well as a substrate level phosphorylation leading to formation of GTP, and all these steps lead to the regeneration of the oxaloacetic acid, which starts the citric acid cycle. Now here the continuous supply of oxaloacetate is always necessary for the running of the Krebs cycle. So this is the overview of the Krebs cycle. Let's read it in more detail with the enzymes associated. So this is the schematic representation of the Krebs cycle, otherwise known as the tricarboxylic acid cycle or the citric acid cycle. Okay. Now here acetyl coenzyme A, the 2-carbon compound which is formed in the link reaction in the mitochondrial matrix, undergoes a step of condensation with a 4-carbon compound oxaloacetate. As a result of the condensation, 4C plus 2C gives rise to a 6-carbon compound, that is citrate, and subsequently a molecule of water is added and the coenzyme A is released. The enzyme catalyzing this reaction is citrate synthase. Okay, as citrate is being synthesized, the enzyme is citrate synthase. In step number 2 of Krebs cycle, citrate is getting isomerized to isocitrate, which are both 6-carbon compound. Now, the enzyme catalyzing it is aconitase, and it causes a dehydration step first, that is removal of hydroxyl group, and subsequently a reattachment of hydroxyl group takes place, leading to the formation of isocitrate. Now, why this dehydration and rehydration is necessary? This dehydration and rehydration step ensures that the OH group is attached in isocitrate in such a way that it is in vicinity with the carbonyl group, as a result of which the OH group can be oxidized further and the carbon dioxide can be easily removed, or decarboxylation can take place easily in isocitrate. So this step of oxidative decarboxylation takes place when isocitrate gets converted into 5-carbon compound alpha ketoglutarate in step number 3. So step number 3, there is an oxidation of the isocitrate first, and the OH group is oxidized. Subsequently, a molecule of carbon dioxide is released, leading to the formation of the 5-carbon compound alpha ketoglutarate. Now, when isocitrate is getting oxidized, what should be getting? There should be something which should be reduced. So, what is getting reduced? Here, NAD plus is getting reduced to NADH plus H plus. Okay. Now, the enzyme here is isocitrate dehydrogenase. Okay. Now, in step number 4, it is similar to step number 3. Now, what happens here is alpha ketoglutarate, which is a 5-carbon compound, undergoes an oxidation first, and then subsequently a molecule of carbon dioxide is released, leading to the formation of 4-carbon compound succinyl coenzyme A. The enzyme catalyzes the reaction is alpha ketoglutarate dehydrogenase. Okay, here also while alpha ketoglutarate is being oxidized, NAD plus is getting reduced to NADH and H plus. Is that clear? So here you should remember that whenever NADH comes into picture or FADH2 comes into picture, the enzyme which should be catalyzing the reaction is always dehydrogenase. Okay, so here isocitrate is getting converted, so isocitrate dehydrogenase. In step number 4, alpha ketoglutarate is getting converted, so the enzyme is alpha ketoglutarate dehydrogenase. Right, is that clear? Now next, let's see step number 5 of Krebs cycle. So the succinyl coenzyme A formed, which is a 4-carbon compound, loses it, coenzyme A group, and the product form is succinate. So here the succinyl coenzyme A also is, leading to the formation of the GTP, which is nothing but guanosine triphosphate, comparable to the ATP. So first substrate level phosphorylation takes place when succinyl coenzyme A is getting converted to succinate and losing its coenzyme A group. Is that clear? The enzyme catalyzing the reaction is succinyl coenzyme A synthetase. Remember, in case of enzymes, whenever an ATP is formed, instead of A's, we end the enzymatic name with tase. So succinyl coenzyme A synthase. Instead of that, we will write succinyl coenzyme A synthetase. Okay. Next, let's see in step number 6 what's happening. The succinate, which is a 4-carbon compound, leads to the formation of fumarate, which is again a 4-carbon compound. But succinate is getting converted that time when oxidation step is taking place. So succinate is getting oxidized to fumarate, and the subsequently FAD plus is getting reduced to FADH2. Okay, so this enzymatic step is catalyzed by succinate dehydrogenase. Okay, so here you should remember that FADH2 is also a redox equivalent, which is comparable to NADH. Okay, and this is enzyme is very important because the succinate dehydrogenase is the only enzyme in the Krebs cycle that is present in the inner membrane mitochondria. All the other enzymes of the Krebs cycle, they are present in the mitochondrial matrix, excluding succinate dehydrogenase, which is present in the inner membrane of mitochondria. Okay, so let's come back to Krebs cycle now. So fumarate, which is formed, is a 4-carbon compound. It is accepting a molecule of water, and hydration takes place, leading to the formation of malate, which is again a 4-carbon compound. Conversion of fumarate to malate is catalyzed by fumarase enzyme, and in the final step of Krebs cycle, oxaloacetate is regenerated from malate. So malate undergoes oxidation, leading to the formation of oxaloacetate, and in the subsequent process your NAD plus is getting reduced to NADH and H plus. So because the dehydrogenation takes place here, the enzyme is malate dehydrogenase. Okay, so if you see, we are getting two types of redox equivalents here. So 3 NADH plus H plus we are getting. Along with that, we are getting one FADH2 in the Krebs cycle. Okay, so 3 NADH and 1 FADH2 in the Krebs cycle for a single molecule of acetyl coenzyme A or a single molecule of pyruvate. In glycolysis, but how many pyruvates we have? In glycolysis, we will be having 2 pyruvate. So how many total NADH we will be getting? If for one pyruvate we are getting 3 NADH, for a single glucose we are having 2 pyruvate, that means we are getting 6 NADH in Krebs cycle. Similarly, FADH2, how many we will be getting? If for single pyruvate we are getting 1 FADH2. So for 2 pyruvates or a single molecule of glucose, we will be getting 2 FADH2. Apart from this redox equivalence, which will help us in getting the ATP indirectly, we are getting ATP synthesis directly here, or substrate level phosphorylation as indicated in the formation of GTP. So 1 GTP is produced per Krebs cycle. So for glucose, we will be needing 2 tons of Krebs cycle. So we will be getting 2 GTP or 2 ATP by substrate level phosphorylation. So that's all about the Krebs cycle. I hope you got clarity about it. Let us now calculate how many ATPs are produced in the link reaction as well as the your Krebs cycle. Okay, so we know that in glycolysis a single molecule of glucose is forming two molecules of pyruvate. In the link reaction, single molecule of pyruvate is being converted into acetyl coenzyme A, one molecule, and it leads to the formation of one NADH. Right, no ATPs are produced. So if we consider glucose, so two pyruvates are formed, so which means two acetyl coenzyme A and two molecules of NADH. Okay, so two molecules of NADH, they come from the link reaction. Okay, now let's see in the Krebs cycle. So one molecule of acetyl coenzyme A enters the Krebs cycle, and here three molecules of NADH are produced, one molecule of FADH2 is produced, one substrate level GTP or ATP, one molecule of ATP is produced. Also, two molecules of carbon dioxide are produced here. Okay, so when we see one molecule of glucose, it means that two acetyl coenzyme A will be formed or two turns of Krebs cycle will be required. Okay, so NADH, how many NADH we will be getting? So 3 cross 2, that means 6 NADH. 1 cross 2, that means 2 FADH2, 2 molecules of GTP and 4 molecules of carbon dioxide we will be getting. So let us now calculate the number of ATPs. So we know that a molecule of GTP is equivalent to 1 molecule of ATP. Now NADH, one molecule of NADH leads to the formation of 3 ATP molecules. So in total from the link reaction we would be getting 2 cross 3, that is 6 molecules of ATP we would be getting. Now, one molecule of FADH2, it leads to the formation of two molecules of ATP. So, 2 cross 2, that is 4 ATPs will be produced by FADH2. So, if we see the if we see the Krebs cycle, total we are getting 18 plus 4 plus 2, that is we are getting 24 ATP. Now, if we consider the link reaction also, so where we are getting 6 ATP in total, link reaction plus the Krebs cycle, we are getting 6 plus 24, that is 30 ATPs. Okay, now let's see the carbon dioxide. So in link reaction, one decarboxylation process was taking place. So for a single pyruvate molecule, we were getting one carbon dioxide, and for one molecule of glucose, we have two pyruvate or two carbon dioxide. So in total, we are getting one carbon dioxide from the link reaction which is released, and two carbon dioxide from the Krebs cycle which is released. So we can rewrite the equation of respiration, aerobic respiration, like this: pyruvic acid plus 4 NAD plus 3 from Krebs cycle, 1 from link reaction, 1 required for link reaction, plus 1 FAD plus 2 molecules of water plus ADP, it gives rise to 3 molecules of carbon dioxide. 1 is released in link reaction, 2 is released in Krebs cycle, and leads to the formation of 4 NADH, 3 in Krebs cycle, 1 is in the link reaction, 1 molecule of FADH2 and a single molecule of ATP. Is that clear? Is this reaction clear? Now, if you want to write it for glucose, you simply multiply it with 2, because in glucose we get 2 molecules of pyruvate. Okay, I hope this calculation of ATP is clear for you guys. So in last slide, we studied about the the Krebs cycle, link reaction and the glycolysis. Now we will see the final part of the respiratory system, that is the electron transport system. So it is in this step that the oxygen is being used and it is converted to water. Now here, since oxygen is used in this step, we also call this process oxidative phosphorylation. So what essentially happens in ETS is that the NADH and FADH2, which are the redox equivalents that are produced in the Krebs cycle as well as in the link reaction and glycolytic pathway, they come and enter the membrane, the inner membrane of mitochondria, and they lead to the formation of the ATP. So in ETS, indirect formation of ATP is achieved with the help of the proteins that constitute the electron transport system, or ETS. Typically, there are five proteins, that is sub that is 1, 2, 3, 4, and 5, that are responsible for generating the ATP and are present in the inner membrane of mitochondria. Is that clear? Now, how is this ATP produced? The concept is, the this is the mitochondria structure, this is the outer membrane, this is the inner membrane, and in the inner membrane we have the proteins, or the components of the electron transport systems that are present. So these ETS systems, they act in such a way that the inside of the mitochondria, or the matrix, the inner space or the matrix of the mitochondria becomes deficient in the proton, or H plus ion is less in the matrix as compared to the inner membrane space, or the IMS. So our goal is to increase the proton concentration in the IMS and decrease the proton concentration in the matrix. So this proton the difference in the proton concentration in IMS and matrix, it sets up a proton gradient, and this proton gradient leads to increased H plus in IMS and decreased H plus concentration in matrix. So there will be a tendency of the protons to flow from the from the inner membrane space towards the matrix, and this proton gradient leads to the formation of ATP. So that is the concept for the electron transport system. Okay, and how ATP is being generated. Now, next concept which you need to understand is, how is this proton gradient achieved? Right. Now this proton gradient is achieved from the movement of electrons across the different redox centers. So what are the different redox centers? They are nothing but the component of the electron transport system, or the components of the ETC, right? So the movement of electrons across the electron transport system causes the proton gradient to build up. As the electrons become, as the components of electrons transport chain receive the electrons, they become excited, and a molecule does not want to be in its excited state. So that excited component of ETC, components of ETC, or the excited redox centers present in the ETC, when they come to the de-excited state or their ground state, they release the energy. So this energy released during the de-excitation of the components of electron transport chain, they help or give the energy to pump the protons from the matrix to the IMS, and this helps in the development of the proton gradient. In the final step, the proton gradient is utilized to make the ATP. Is that clear? So now the components which help in the maintaining the proton gradient are component 1, 2, 3 and 4, while the fifth component is responsible for the ATP synthesis, or it is the ATP synthase component. Okay, another concept which you need to understand is who gives the electron. Where does the electron comes from? Okay, the electrons, it accepts the electrons, becomes excited. When it comes back to the ground state, energy is released, that is helped to maintain the proton gradient, and proton gradient is utilized to make ATP. Next question which comes is who gives the electron. So here NADH and FADH2 are the redox equivalents, or the molecules which give the electrons. Right? So how do they give electrons? We have seen that NADH was formed when NAD plus uh, at, uh, accepted a hydride molecule. And what is again a hydride molecule? I have already told. It means a proton, when it accepts two electrons, it forms the hydride molecule. So when NADH comes in vicinity of the enzymes of electron transport system, it loses the hydride ion. In turn, it loses the two electrons. These two electrons are accepted by the components of the electron transport chain. Similarly, FADH2 also becomes oxidized and becomes FAD plus and releases its H2. And what is this H2? That is again 2H plus 2 electrons. So that is how the electrons are accepted or obtained by the components of the electron transport chain. Okay. Now, next question is, why do the components of the ETC, they accept the electrons? It is because the components of the electron transport chain have different redox potential, and they have a tendency towards accepting electrons, or they have their affinity towards accepting the electrons. They have an affinity towards the electrons. So the electrons are accepted by the simultaneous release of electrons by NAD plus, right? Unless someone is willing to accept the electrons, you cannot lose them, right? Because redox centers in ETC are willing to take up those electrons. That is why NADH is able to release the hydride ions or release the electrons. And how is the movement of electrons possible? The movement of electrons is possible because the redox centers are arranged in such a way that they have they have a difference in affinity towards the electrons. So the if you come towards the like component 4, they would be having a greater affinity towards the electrons. So the movement of electrons is always in a direction in such a way that it transports from 1 through 4. Okay. Finally, the electrons are accepted by the oxygen, which is getting reduced to water, and oxygen acts as a final electron acceptor, and water is produced. Okay. So in the fourth subunit, oxygen comes as a major player and the final acceptor of the electrons, and the proton gradient which was hence formed is helping in releasing the ATP. I hope the concept about ETS is clear. Now let's see what are the individual components of ETS. Okay. So this is the schematic representation of the different components that are present in electron transport system. So to revise, we saw that all the components of the ETC, where are they present? They are present in the inner membrane of the mitochondria. So consider this as the outer membrane of the mitochondria. This is the inner membrane of the mitochondria. Inside it, we have the matrix, and in between the two membranes, we have the inner membrane space, or the IMS. Okay, so now what do we need? We need the proton concentration to be less in the matrix and to be more in the IMS. Is that clear? So now the NADH and FADH2 will be helping in releasing the electrons. So let's see how it happens. So before that, let us see what are the different complexes in ETS. So I told in ETS there are 5 complexes, and the 4 complex 1, 2, 3, 4, they help in movement of the electrons, while complex 5 is the ATP synthase unit F, which is the F0 and F1 unit, just like as we saw in the photosynthesis. So this complex 5 helps in just making the ATP through the proton pumping, which happens, and it gets its energy from the proton gradient, hence releasing the ATP. Okay, now complex 1 is called as the NADH dehydrogenase complex, and it is made up of FMN and iron sulfur unit. Okay, and also ubiquinone is present. Okay, ubiquinone is also called as a coenzyme Q. Now complex 2, it is also called as the succinate dehydrogenase complex, and it is made up of FAD and your FES or iron sulfur unit. Now next you have the complex 3, which is the cytochrome BC1 complex, and it also has an iron sulfur center in it. Now complex 4, which helps in the reduction of the water, is also called as a cytochrome C oxidase complex. It is important that it has 2 copper centers in it, copper center A and copper center B. Along with that, it has molecules like cytochrome C, cytochrome Q and A3. Okay, cytochrome C, A and A3. So these are present in the complex 4. Now what happens, now we know that about the complexes, the names of it. So you should see that the protons, they are pumped by only the complex 1, 3 and 4, but complex 2 does not pump protons. Why? I will come that to it a little later. Next thing which you need to remember, that NADH gives its electrons through complex 1, which then travels through complex 3 and 4. So complex 1, 3 and 4 are involved in the movement of electrons which are released by NADH. Okay, and where does the FADH2 give its electrons? FADH2 gives it electrons to complex 2, and it passes through complex 3 and complex 4. Okay, so now the complex 3 and 4, they are common in both FADH2 and NADH, and they receive electrons from both of them. Okay, now that you know about all these things, let us see how it works in a little more detail. Okay, now see, in matrix you will have the NADH. NADH is having a hydride ion, and proton plus always comes because to maintain the electron neutral, the charge balance, the neutrality, because NADH is having hydride ion H minus, which is essentially meaning that it has 1 proton and 2 electrons. So in order to balance the charge, we will also have 1 proton associated. Okay, so NADH will have a tendency to give it electrons to FMN. Why? Because FMN will have a tendency to accept the electrons. So it accepts the electrons and transfers it to FES, which is further transferred to ubiquinone or coenzyme Q. Now this ubiquinone is mobile, and it can get reduced to coenzyme H2, and it passes the electrons to complex 3. Now complex 3 gives its electrons to it gets its electron from the reduced ubiquinone, and cytochrome B accepts the electrons from ubiquinone, which further gives it to iron sulfur center and cytochrome C3. Is that clear? Now, this cytochrome C3 gives it in the complex 3 gives its electron to cytochrome C. Now, cytochrome C is a mobile electron carrier and it shuttles the electrons from the complex 3 to complex 4. Now, cytochrome C can travel through the membrane. It comes here and gives its electrons to the copper center A. Now, this copper center becomes activated and it does not want to be in high energy state and it passes its electron to copper center B via cytochrome A3. Okay, now the finally the electrons are accepted by an atomic oxygen or half O2 and it is reduced to water. Okay, so that's how the electrons from NADH move from complex 1 to 4 via complex 3. Okay, and in the process, finally electrons are accepted by water, accepted by oxygen, releasing the water molecule. Okay. Now let's see what happens to FADH2. Now in FADH2, I already told that what accepts the FADH2? It is complex 2. Now complex 2 accepts the electrons from FADH2 and it gets oxidized. FAD becomes oxidized and it passes on its electrons to iron sulfur center, which further passes on to ubiquinone, reduces it, and this ubiquinone which is reduced passes its electrons to cytochrome B, FES, cytochrome C3. Again this cytochrome C3 passes the electrons to the mobile electron carrier, that is cytochrome C, which again gives its electron to copper center B via copper center A, cytochrome A and A3. Okay. Finally, electrons from FADH2 are accepted by the by the oxygen and water is being formed. Okay, so oxygen here acts as the final acceptor. So the electrons which are released from FADH2 are moving through complex 2, then 3 and 4. So for NADH2, complexes involved are complex 1, complex 3 and complex 4. For FADH2, complex 2, complex 3 and 4 are utilized. Okay. So we saw the movement of electrons. Now let's see how the protons are being pumped. So for that I told that you should remember complex 2 does not pump the protons. So how does it pump the protons? So here you see the complexes are nothing but the proteins. Now the proteins are of complexes 1, 3 and 4 are arranged in such a way that they have a proton channel in them. So with the movement of the electrons, whatever energy is utilized as the excited redox centers become de-excited, that energy is utilized to pump the protons through what? The proton channels from the matrix towards the inner membrane space. So for each molecule in NADH, when it passes through complex 1, 4 protons are being pumped to the IMS. Similarly, when the NADH is passing through the complex 3, 4 protons are again pumped here. And in complex 4, 4 should have been pumped, but 2 protons are utilized to reduce the oxygen. So net gain is 2 protons which are released to the complex 4. So if you see the NADH2, it is able to pump 10 protons to the IMS. Okay. Now let's look at FADH2. Now FADH2, I told it enters from complex 2. So it is complex 2 is not having any proton channel, but it promotes the movement of the proton in the other complexes, that is 3 and 4. So as it gives the electrons to the complex 3, complex 3 has proton channel in it, so it can pump 4 protons from the matrix towards the IMS. Similarly, in complex 4, 2 are being used to reduce the atomic oxygen. So 2 protons are pumped to the inner membrane space. So FADH2 in total it is generating 6 protons. Is that clear? It is a very important concept. I hope you are clear with it. So net result, when we come to the complex 5, what do we have here? If we see IMS and the matrix, the concentration of the protons is very high here in the IMS compared to the matrix. So the protons will tend to flow from the IMS towards the matrix through F0 and F1 complex. F0 is an integral protein and acts as a transmembrane channel, and F1 is the ATP synthase unit. So F0 acts as a rotor and leads to the formation of the ATP through the headpiece from ADP. So ATP is being released, which is our ultimate motive in the complex 5 of the ETS. Okay, I hope you are clear about this concept. Two more things you need to remember. You see, what are the molecules which are able to switch from one complex to other? You see the ubiquinone, which is present in the inner membrane, and it can switch from complex 1, it is giving to complex 3, and again from complex 2 to complex 3, right? So ubiquinone is a mobile electron carrier and is present in the inner membrane. And what about cytochrome C? That is very, very, very important. Now cytochrome C is also a mobile electron carrier and it is present in the outer membrane, in the outer part of the inner membrane towards the inner membrane space, and it can shuttle the electrons between complex 3 and complex 4. Okay, so these are all the things you need to remember. So these all points we already covered, but just let's revise it. So the energy stored in NADH plus H plus and FADH2 is passed on to oxygen, leading to formation of water. And the the electrons from NADH produced in the mitochondrial matrix during citric acid cycle is oxidized by NADH dehydrogenase complex, and final acceptor in complex 1 is the ubiquinone. Okay, so ubiquinone receives the reducing equivalence via both NADH and FADH2 and it is receiving the electrons from both complex 1 and 2. Okay, the reduced ubiquinone is oxidized with transfer of electrons to cytochrome C via the cytochrome BC1 complex. And cytochrome C is a small protein attached to the outer surface of the inner membrane and acts as a mobile carrier for transfer of electrons between complex 3 and 4. Okay, so all these points are important. After you understood the concept, you can just remember the names of the ETS components as well as their functions. So oxygen here acts as a final hydrogen acceptor, and the energy of oxidation reduction is utilized to make the ATP. Because oxygen is used to make ATP, this process is called as oxidative phosphorylation. Unlike your photosynthesis, in which light energy was used, we were calling it photophosphorylation. We call it oxidative phosphorylation because ATP is synthesized with the help of the oxygen. Now let's see the complex 5. The complex 5 is acting as the ATP synthesis unit. So it has two components, F0 and F1. F0 is the integral protein, F1 is the extrinsic protein. It is on the matrix side, and F0 is the transmembrane, acts as the transmembrane channel, and it is in the inner membrane. Okay, so as the proton gradient is established, it forms the proton motive force, and it helps in the movement of F naught, just like a motor, just like a rotor. And from the head piece, ADP and phosphate are utilized to form ATP towards the matrix side. So let's see how many ATPs are produced from NADH. So roughly about 3 ATPs are formed from NADH. So it is mentioned that for each 4 hydrogen plus or 4H plus ion, ATP is produced, but approximately about 2.7 to 4H plus are required to make a molecule of ATP. So check NADH, we saw 10 H plus are produced from NADH, and 6 are produced from FADH2. So if we calculate, approximately 3 protons are utilized to form one molecule of ATP. So net gain is 3 ATPs that are produced here. Similarly, in FADH2, we saw 6 protons are produced. So we will be getting 3 from 3 protons, we are getting 1 ATP. Another 3 protons, we are getting another ATP. So we are getting 2 ATP from FADH2 while we are getting 3 ATPs from NADH. I hope the synthesis of ATP part is clear for you guys. Now let's just quickly revise the respiratory balance sheet. So what's happening here is, this is your respiration process and this is the glycolysis. In glycolysis we saw that 4 ATPs are produced and 2 ATPs are utilized. So net gain of ATP is 2. And 2 NADH were also produced, which accounts for 6 ATP. So total in glycolysis we are getting 8 ATPs. Now check the link reaction. In link reaction we don't get any direct ATPs, we just get 2 NADH. Remember, 1 NADH per pyruvate. So 2 NADH for 2 pyruvates or for a single glucose molecule. So 2 NADH means 2 cross 3 or 6 ATPs. Now check the Krebs cycle. We have already discussed it, but again we will just quickly revise it. So you have direct ATP and indirect ATP. So we got 2 direct ATPs or in the form of GTP, and we got here for indirect we had 6 NADH, 2 FADH2, so total 22. So if you calculate 22 plus 2, it makes 24 ATP. So if you combine all, in glycolysis we have 8 ATPs, link reaction 6 ATPs, and Krebs we have 24 ATP. So total we should get 38 ATP. But this maximum possible ATPs, that is 38, is achieved only in the bacteria. In eukaryotes we have general gain of 36 ATPs, which because 2 ATPs are utilized in pumping. Okay, so you should remember while calculating ATP that ATP we have to see from 2 sources, direct as well as indirect. Direct is from substrate phosphorylation, which is either a GTP or ATP synthesis, and indirect is from the oxidative phosphorylation or ETS via the oxidation of NADH or FADH2. Now this gain of 38 or 36 ATPs is obtained only when we have few assumptions, that is the respiratory process, it happens in a very sequential or orderly pathway, and this NADH is synthesized in glycolysis, is transferred into mitochondria and undergoes the oxidative phosphorylation, and none of the intermediates of the pathway are utilized to synthesize any other compound. So under ideal condition, only when the glucose is being respired and there are no other alternative substrates, we will be getting this number of ATPs in the maximum possible case. Now let's see what type of process is this respiratory process. Now respiration, we already saw that there is breakdown of the sugar molecules or catabolism is taking place, but it can also act as an anabolic pathway in which new products are formed. So see, this acetyl coenzyme A, which is an intermediate in the Krebs cycle, can be utilized for fatty acid synthesis. Similarly, oxaloacetate, ketoglutarate, succinyl coenzyme A, they can be used for the protein synthesis, and the glyceraldehyde 3-phosphate and fructose 6-phosphate can be used in the carbohydrate synthesis. So you see, respiration is accounting both the catabolic and the anabolic pathway. So together it can be very correct to say that respiration is an amphibolic pathway because it has both catabolism and anabolism in it. Now, this is a chart which shows that, in the respiration, different substrates can act as a, for the respiratory substrate, such as fat, carbohydrate, and proteins. For fatty acids, directly acetyl coenzyme A will be produced, which can act as the entry point for the Krebs cycle. So, in all of these three cycles, Krebs cycle is the common respiratory pathway, and fats, carbohydrate, and proteins, all of them can act as a respiratory substrate. But ideally, carbohydrate is the most commonly used. Now let's come to the final part of the respiration subunit, respiration unit, that is the respiratory quotient. Now what is respiratory quotient? Respiratory quotient is how much volume of carbon dioxide which is evolved in a reaction divided by how much volume of oxygen that is consumed in a reaction. So, depending upon the different substrates we use, or depending upon the type of the respiratory substrate, we can have different RQ values. Now, if you see carbohydrate, for 6 molecule of carbon dioxide that are produced, 6 molecule of oxygen is used. So, RQ becomes 1 here. Now, if you take proteins, RQ is generally 0.9. It is less than 1. Fats also have an RQ which is less than 1, and generally it is about 0.7. Organic acids are the compounds which have RQ value more than 1. Example, malic acid, it will have a RQ value of 1.33, and oxalic acid will be having a very high RQ value of 4. So if you arrange the RQ values of different respiratory substrate, so you have oxalic acid followed by malic acid followed by carbohydrate followed by protein and finally fats. Okay. Now let's calculate what would be the RQ value for a fat that is tripalmitin. You see 102 carbon dioxide units are produced divided by 145 oxygen which is utilized, giving the net 0.7 RQ for tripalmitin. Okay, if the question comes like that, it would be very easy for you to calculate. Now, let us see what is the RQ in case of fermentation. So, fermentation is an anaerobic process. No oxygen is used and RQ hence becomes something by 0. It becomes infinity. Okay. Now, if we see the fatty seeds, okay, fatty seeds, if we see the maturing condition in which fatty acids are synthesized, lot of, a lot of active biosynthesis leads to more carbon dioxide which is produced. So RQ value becomes more than 1. Why? Because carbohydrates are converted to fat, and in this process this active biosynthesis leads to more carbon dioxide with respect to oxygen. So RQ value becomes more than 1. Now if you see the germinating seeds, in germinating seeds fat are being used as the main fuel. So when fat are used as the main fuel, the RQ value, it becomes, you see, for when fats are used, what is the RQ value? It is less than 1. So as fats are used as the main fuel, RQ value becomes equal to almost equal to 0.7. Is that clear? Next, let's see the succulents. If you see succulents like Opuntia, which have a CAM pathway in them, and during night condition, if you see these succulents, they thrive in very dry condition, and they have, they thrive in very dry condition, and they have the stomatals which open only during the night time. So during the night time, the stomata is open because they don't want any loss of water molecules or carbon dioxide, so they remain closed in the daytime and open at the night time. So when in the night time the stomata is opened, the carbon dioxide which is released in the respiratory pathway, incomplete conversion takes place. Carbon dioxide is evolved, is utilized to produce the organic acid such as malic acid, which will be utilized during the daytime to release carbon dioxide and carry out photosynthesis. So as the carbon dioxide almost become negligible or zero because it is utilized in formation of organic acid, that is malic acid, so RQ in succulents in night becomes equivalent to zero, especially in case of plants such as your Opuntia. In others, it becomes very minimal. If you say in the time of day, carbon dioxide, the stomata is open, whatever carbon dioxide is utilized, it is released during the conversion of malic acid that is used for photosynthesis, and the oxygen is also consumed in the process of respiration. So in total, what happens that you get RQ value almost equal to 1 in case of the succulents during the daytime. Okay, I hope you are clear about the respiratory quotient. I hope you are clear with the different concepts we have discussed in this chapter today, that is respiration in plants. So to read this chapter, understand the concepts first in the chapter which are there in your glycolytic pathway, Krebs cycle, link reaction, okay, and ETS. And after understanding, you might want to remember the remember the names and how much ATP is produced, right, or an RQ values. First understand the concepts and then remember. That is how you can read this chapter, okay? And I hope you like the session today. So please subscribe to my channel, share the video, and hit that like button. Thank you everyone. Have a very good day.